vetenskapsakademin och den här presskonferensen då vi ska presentera årets Nobelpris i kemi. Welcome to the Royal Swedish Academy of Sciences and this press conference when we will present this year's Nobel Prize in Chemistry. We will keep to our tradition and begin the presentation in Swedish and then continue in English. And you're of course welcome to ask questions in either language later on. Jag heter Hans Sellegren och är ständig sekreterare här på Kungliga Vetenskapsakademin. Och till höger om mig sitter professor Heiner Linke, ordförande i Nobelkommittén för kemi. Och till vänster om mig sitter professor Johan Åkvist, ledamot av Nobelkommittén för kemi och expert inom ämnet. My name is Hans Ellegren. I'm the Secretary General of the Royal Swedish Academy of Sciences. And to my right is Professor Heiner Linke, Chair of the Nobel Committee in Chemistry. And to my left, Professor Johan Åkvist, a member of the Nobel Committee for Chemistry and expert in this field. Årets pris handlar om livets kemiska verktyg, proteinerna. This year's prize is about the chemical tools of life, proteins. Kungliga Vetenskapsakademin har idag beslutat att tilldela 2024 års Nobelpris i kemi med ena halvan till David Baker, University of Washington, Seattle, USA, för datorbaserad proteindesign. Och andra halvan gemensamt till Demis, Hassabis och John Jumper, Google DeepMind, Storbritannien, för proteinstrukturprediktion. The Royal Swedish Academy of Sciences has today decided to award the 2024 Nobel Prize in Chemistry with one half to David Baker, University of Washington, USA, for computational protein design, and the other half jointly to Demis Hassabis and John Jumper, Google DeepMind, United Kingdom, for protein structure prediction. Professor Heiner Linke will now give us a short summary in English. Please, Heiner.
We have a great prize for you today. Proteins are the molecules that enable life. Proteins are building blocks that form bones, skin, hair, and tissue. Proteins are motors that power our muscles. They are the machines that read, copy, and repair DNA. They are the pumps that keep our neurons in our brain ready to fire. They are antibodies that enable our immune response, sensors that our cells use to communicate, and hormones that regulate all the cells in our body. A protein consists of hundreds or thousands of atoms. The protein's biochemical function is determined by the precise position of each of these atoms in space relative to one another. To understand how life works, we first need to understand the shape of proteins. Therefore, it has long been a dream to learn to predict the three-dimensional structure of proteins from knowing their amino acid sequence, which is encoded in the DNA. For several decades, this was considered impossible. 4 years ago, in 2020, Demis, Hassabis, and John Jumper managed to crack the code. With skillful use of artificial intelligence, they made it possible to predict the complex structure of essentially any known protein in nature. Another dream of scientists has been to build new proteins to learn how to use nature's multi-tool for our own purposes. This is the problem that David Baker solved. He developed computational tools that now enable scientists to design spectacular new proteins with entirely novel shapes and functions, opening endless possibilities for the greatest benefit to humankind.
Thank you, Professor Linke. And Professor Åkvist, are you ready to give a more detailed presentation? Yeah.
So proteins, they are strings of amino acid molecules. And these strings, what they do is that they fold themselves up into complicated patterns in three-dimensional space. So here we see the structure of a small protein with all its atoms depicted. Now, this is difficult to understand, so we use these simplified representations with ribbons and arrows to show how this chain is folded in space. Now, the most remarkable thing here is that this three-dimensional shape is actually encoded in the sequence of amino acids. So this was the subject of the 1972 Nobel Prize in Chemistry to Christian Anfinsen, who made this discovery. So if the three-dimensional structure is encoded in the sequence of amino acid, it means that we should be able to predict this three-dimensional structure just from the sequence of amino acids. And this is what protein structure prediction is all about, to use the sequence to predict the three-dimensional structure. Now, we can also turn the problem around and ask, if I draw a structure here, what would be the amino acid sequence that would give this structure? For example, I could draw a structure that nobody had ever seen before, a completely new protein, and then try to find a sequence that would give this protein. Then I could go to the lab, I could make this sequence, and thus I could make completely new proteins that have never been seen before in nature. So this is what protein design is all about, creating completely new proteins. So this year, we're awarding discoveries in these two areas, protein structure prediction and protein design. And as you will see, they are tightly knit together. Now, the design problem was solved first. In 2003, David Baker and his colleagues drew this protein, which had never been seen before. There existed nothing like it in nature. And then they had written a fantastic computer program called Rosetta that tried to find a sequence that would give you this structure. So here comes the sequence that Rosetta typed out. You don't have to remember it, but it's written in the one-letter amino acid code. So they went and made the protein, and then they used X-ray crystallography to determine what it looked like. And it turned out to look almost exactly as predicted. So this was the big breakthrough in computational protein design. And the Rosetta program basically works by picking shorter fragments from a database and piecing them together, optimizing their sequence and their structure. And this was the proof that protein design, computational protein design, really worked. Since then, the applications have been very many, and David Baker opened up a completely new world of protein structures that we had never seen before. Here you see some examples of the very many that have been published. You see, for example, inhibitors of the COVID virus spike protein, these yellow things to the upper left. He designed a fentanyl sensor that will allow you to detect fentanyl in the environment. He has made nanomaterials, also for the purpose of vaccine development. It's a mind-blowing development, and it's almost as if it's only your imagination that sets the limit for what you can do here. Okay, back to protein structure prediction. The other half of the prize. Now, this is a very difficult problem, because even for a moderate length amino acid sequence, let's say 100 amino acid residues, there are an astronomical number of possible structures. So it's clear that you need computers to try to solve this problem as well. Now, the accuracy of protein structure prediction was pretty low for many years. So you can see from this graph, up until 2016, it had barely reached 40% accuracy. So this field was moving slower. But then something happened in 2018, and particularly in 2020, when the program AlphaFold 2 was published. It was published the summer of 2021. The results came in 2020. So this work was led by Demis, Hassabis, and John Jumper at the DeepMind company. And AlphaFold 2, which had a predecessor called AlphaFold 1, that already was a step forward. But AlphaFold 2 reached now almost 90% accuracy. So this started to be compatible with experimental errors in structure determination. So it was an enormous breakthrough. How did they do it? Well, they used neural networks and deep learning. So for those of you who were here yesterday, you know all about this. And AlphaFold 2 is an ingenious piece of neural network design. I will briefly try to tell you a little bit about how it works. So it makes use of sequence databases. There is a database of all known protein sequences, about 200 million sequences. There is a database of protein structures. Those are only about 200,000 today. But the program is trained on these databases, and you give it an input sequence, and then the program does, or the network does two things. It creates an alignment of similar sequences from different species, for example. And these could be thousands of similar sequences that it finds in the database. Now, what it does, it looks for correlations in these sequences. So if you have a mutation in one position, maybe it's correlated with a mutation in another position. And from this information, it can make a distance map, as we see in the lower picture here, which basically tells you how close are two amino acids to each other in space. So the sequence alignment contains information about such distances. And then these two objects, they update each other continuously as the network does its job until it comes up with a final distance map. And then the program uses an ingenious method to convert this distance map to a three-dimensional structure with 90% accuracy. So protein structure prediction with AlphaFold 2 caused a complete revolution in structural biochemistry. It has over 2 million users at present. And the AlphaFold team immediately made a database with predicted structures for all the 200 million sequences that we know today. So this is a fantastic resource for biochemical and biological research. So there have been numerous applications of AlphaFold. You can, for example, mine this database for new enzymes that, for example, degrade plastics. You can also use AlphaFold for solving huge structures. So the one you see on the right here is one of the largest molecular structures known in biology. It contains over 1,000 proteins. So they could solve this structure by combining AlphaFold with electron microscopy data. So the impact of this year's prizes in protein design and protein structure predictions are truly huge, and the results are spectacular. So I would just leave you with a message that in order to understand how proteins work, you need to know what they look like. And that's what this year's laureates have done. Thank you.
Thank you, Professor Åkvist. We shall see now if we have one of the laureates. Professor David Baker, are you there? Good morning, and please accept our warmest congratulations to receiving the Nobel Prize in Chemistry.
Thank you very, very much. Deeply honored.
I must ask, how do you feel right now?
Very excited and very honored.
Yeah, I think that's very well deserved, actually. I'm sitting here in this beautiful session hall of the Royal Swedish Academy of Sciences, and here at this press conference, there are many interested journalists, both from the Swedish and the international press. Would you be ready to take some questions from them?
Yes, I'd be happy to.
Sveriges Television.
Thank you. My warmest congratulations to your achievements and to this year's Nobel Prize. My name is Susan Ritz and I work for Swedish Television, and I know our viewers are really excited about how you cracked the code of creating new protein and what inspired you to pursue this truly groundbreaking research.
Well, I stood on the shoulders of giants. My colleagues Steve Mayo and Bill DeGrotto had already shown the signs that protein design was possible. And I also have had throughout my career absolutely wonderful colleagues to work with. Brian Coleman came to my group and we started working on protein design. And that led to Top 7. So I would say I've been really deeply inspired by the others in the field and by the people I've worked with.
Thank you.
More questions from the audience here? Yes, please.
Hello, Bogomir Rajewski from the Polish television. Congratulations, Professor. The thing that we're all very excited here, obviously, is the connection to yesterday's prize in physics, and that connection are neural networks and deep learning and, well, basically artificial intelligence and its role in modern science. How would you describe your opinion about the importance of the development of AI for the future of sciences, given your discoveries? Thank you.
Yes, I think it's tremendous. I and my colleagues have been working on protein design for a number of years, and really with the breakthroughs made by Demis and John on protein structure prediction, really highlighted to us the power that AI could have, and that led us to apply these AI methods to protein design, and that has greatly increased the power and accuracy. So our new AI methods are much more powerful our previous traditional scientific model methods. So I think, and I'm really excited about, I think all the ways in which protein design can now make the world a better place in in health, medicine, and really outside in technology and sustainability.
Yes, please.
Hi, I'm Anneli Megner on Swedish TV4. Congratulations. I would like to know which one is your favorite protein and also what it does in the body and why it's your favorite protein.
Oh, that's a good question. I love all the proteins, so I don't want to pick favorites, but I can tell you about one that we designed during the pandemic that protects against the coronavirus. And I've been very excited about the idea of a nasal spray of little design proteins that would protect against all possible pandemic viruses.
So you, So you mean the fungus-like protein on the coronavirus? Is that the protein you're talking about?
Well, it's a completely designed protein, completely new protein that blocks the coronavirus.
Okay.
More questions? Yes, please, over there in the back.
Hello, Paul Rees from Al Jazeera English. Many congratulations. Could you just set the scene for us? What were you doing when you got the call, and how did it change your day?
Well, I was sleeping when the phone rang, and I answered the phone, and I heard the announcement, and then my wife began screaming very loudly, so I couldn't really hear very well, and the very nice person who was talking to me told me to go somewhere where I could actually listen, and I think it was very, very exciting, and I think it's turning out to be quite a unique, special day.
Any further questions?
I was wondering, when you actually broke the code, what was your feeling at that time?
I think it happened over. It really happened over many years when Brian Coleman, working with me, was able to design the first protein. We kind of imagined that was just designing a protein structure. It didn't have any function, and we sort of imagined what could be possible. And then really over the last 20 years, we've been able to design proteins with more and more complex and powerful functions that can do so many different things. And as we got better and better at that, then the scope of applications became more and more exciting. So it's been sort of this huge opening up of possibilities, because the proteins in nature do so many different things. They mediate all the processes in our bodies and in all living things. And we sort of glimpsed at the beginning that it might be possible to create a whole new world of proteins that could address a lot of the problems faced by humans in the 21st century. And now it's becoming possible as we're able to design proteins that can help in so many different areas.
Yeah, final question here.
Yeah, so another question, because I'm wondering, were you surprised actually by the decision? Because in the predictions for Nobel Prizes this year, you were mentioned as a strong candidate for the Nobel Prize in Physics, given the AlphaFold's reception in recent years. So did it come as a surprise for you, or were you expecting a fall from the Nobel Committee today?
It was a surprise. I definitely heard the things on the, that people were talking, but still it was very surprised. And I just really want to thank my family, my wife, Hannah Baker, my parents, who I called right after I got the call, Marcia and Marshall, who I'm very excited will be able to come to Sweden with me, and my children, Amanda and Benjamin. And I also want to thank all the amazing people that I'm working with now, and the amazing people I've worked with over the years who really, did all this work and made it all possible, and have been sort of fulfilling the dream of what's possible with protein design.
Okay, this seems to be the last question from the press for you, Professor Baker. Thank you. And once again, our warmest congratulations to you. And we look forward to meet you here in Stockholm in December for the prize ceremony.
Thank you very, very much. I'm deeply honored.
So let's move on to more questions about the chemistry prize or the research involved, or if you want to ask questions to the committee about their work. And again, you can ask questions in either English or Swedish. Please.
Yeah, it's me again, sorry. But I'm actually wondering, given yesterday's announcement in physics and the decision to award that prize to research that's connected to neural networks and machine learning, did you take that into consideration today in the morning when making the final decision about the Nobel Prize in Chemistry in any way?
Professor Linke?
So thanks for that question. Each Nobel Prize needs to stand completely on its own feet, both today and perhaps in particular in the eyes of history looking back. So that is the main consideration. And today's prize is really about a breakthrough in biochemistry. It's the making the connection in both ways between amino acid sequence and protein structure, which then is related to protein function, has been defined that was actually called a grand challenge in chemistry, and in particular in biochemistry, for decades. So it's that breakthrough that gets awarded today.
Yes, I don't see any further hands here. By that, thank you for your interest. We hope to see you back here next week on Monday when the Academy will announce the Sveriges Riksbanks Prize in Economic Sciences to the memory of Alfred Nobel. Thank you.
interview 我。哦，太好了。那个，
Professor Johan Åqvist, member of the committee of the Nobel Prize in Chemistry. Please tell us, what is this prize about?
So this prize is about protein structure. And proteins are huge molecules that are made from amino acids. And it's a particular sequence of amino acid. They are connected in a chain, and this chain folds itself up into spectacular 3D structures. And then the question is, can you determine what these structures look like by using computers? That's what this year's laureates have done.
And what did the laureates get the Nobel Prize for?
Well, so David Baker gets the prize for designing new proteins. So he could draw protein structures in 3 dimensions, and then he had a computer program that could figure out what sequence of amino acids would give you this structure. Then he could make these proteins, some of which, or most of which, never had been seen before and didn't exist in nature.
And
so
what could he do? What could we do with those?
He could use them for designing virus inhibitors, small sensors for opioids, for example, building new types of nanoparticles, nanomaterials. The scope is really limitless, what you can do with
You
this.
said mind-blowing during his
Yes,
speech.
certainly.
In what way?
Well, the number of designs that they have produced and published, the variety is absolutely mind-blowing. It seems that you can almost construct any type of protein now with this technology.
And there were 2 other laureates, right?
Yes. So the 2 other are awarded for predicting the 3-dimensional structure from the sequence. Now there is a huge number of possibilities for any given sequence. So it's an astronomical number of theoretically possible structures. So what they could do is to use an ingenious neural network approach to solve this problem. You give the program a sequence, and then it will produce a structure for you. So the design goes sort of the other way. I design a structure, and it will produce that program will produce a sequence, which I can then make.
And what could all this be used for in everyday life or in future everyday life?
Well, it's used for all kinds of things. So, for example, now the AlphaFold team, they made a database of 200 million protein structures from this. So now you can start mining this database for new things that you had never didn't know.
Could you give an example? How could it affect
Well, people have been searching for enzymes that catalyze new reactions, for example, and also in the design area. I mean, there are numerous applications where you try to design vaccines, new inhibitors of things like viruses.
And all this has to do with artificial intelligence as well, right?
Well, at least the protein structure prediction half of the price, that is based on neural networks. Now, David Baker's achievements, they were based on his programs that came a lot earlier, and they were more classical simulation programs, you can say.
Can you tell us a little bit about this year's laureates? Is there anything particular you would like to tell?
Well, if you're interested in games, Demis Hassabis and his co-workers, they made already over 10 years ago now, they made this computer program that could beat the world champion in Go, which was thought to be impossible. And they also made a fantastic chess program that was the best at the time. So
from chess to proteins, then?
Yeah.
Were you in the room when they got the call? Is there anything you can tell us about the call, maybe?
They were all very happy and somewhat surprised, I would say.
What would you think, to you, what excites you the most about this year's Chemistry Nobel Prize?
Well, I would say that what's most exciting is that this prize has opened up a completely new world of protein structures. First of all, structures that exist, but we didn't know what they looked like. And the other half, I mean, to design proteins that don't exist in nature, but can do all kinds of wonderful things.
Thank you so much, Professor Johan Åqvist, member of the committee of the Nobel Prize in Chemistry. Thank you.
Okay.